Okay, good morning, Sec 1. So today we're going to begin our new chapter for our last week of each BL. So this chapter will be on the theme of systems, and we're looking at transport system in living things. Now, the previous chapter that we just covered was on cells, right? And we know that cells basically is the basic building block, which is a living unit in an organism, whether is it a unicellular organism or whether is it a multicellular organism. All right, so I think there could be one question that you could ask would be, how do cells communicate with each other? How do cells actually take in the food and the nutrients or the water that it actually needs? For example, in the cell, there is the vacuole. All right, so how is the vacuole going to store water? How is a vacuole going to store mineral salts? There must be a process that's involved in getting in allowing the cell to obtain the nutrients that it needs. So this lecture, we're going to be talking about two main transport processes. Now, the second part of this chapter will be covered in 2021. All right, so today we'll be looking at transport processes that occur between cells, and there are predominantly two processes that you will be learning this time around. Okay, so let us begin. Now I'm going to begin by asking you to investigate the following three situations, all right? So the first situation that we're going to investigate would be, let's have a look at this transparent orbis, which is known as a polymer ball. It is transparent, as you can see here when the person takes up the polymer ball. However, these polymer balls are usually found colored like this. So how did it get from something that is so transparent to something that is so colorful? Pink, blue, purple, green, and yellow. So the question that should run through your mind would be, how did transparent polymer balls become colored? The colored dye must have been surrounding the ball. So how did the colored dye particles actually enter the transparent ball to make it colored? Okay, the next question that we're going to be looking at would be this video. So let's watch this video. And as you're watching this video, I would like you to think about some of the questions that I will be posing as you are watching the video. So one question before you watch the video would be, what do you think caused the drop of blue ink, which is going to be dropped at the top part of this water? Okay, cup of glass of water. How did the ink move? That's the first question. The second question would be, in what direction did the ink move? Okay, and I would just like to give you a context. This is actually cold water and this is hot water. The third question that you want to ask yourself would be, was there a difference in the movement, or how fast did it move between the cold and the hot water? Okay, so as you are watching, let's think about that. Alright, so you're going to be pouring cold water and hot water. Now you would notice that the person actually drops blue ink at the top. Notice how the ink actually moves, and how did it move? In which direction did it move? Did it stay on top, or do you notice that it seems to be moving throughout? And look at the speed at which this is moving compared to this. Is there a difference? So you might want to ask, what is the main difference between these two glasses of water? Alright, that's quite an interesting video. So now let's watch the third video, which is the third scenario. Now in this video, you're going to be seeing a person putting 4 different Skittles, okay, on a plastic, on a agar-agar, transparent agar-agar plate. And I want you to observe what is happening to the color of the Skittles and the color of the transparent agar. Okay, you will notice after some time in the time-lapse video, notice that you can see that the transparent agar actually takes the color of that particular Skittles. Now, why is that so? It seems that the color seems to be moving from the Skittles to the surrounding area. Now, what is causing this to occur? Okay, so I've given you 3 scenarios. All right, so now let us think and ponder about it. You may pause the video to actually write down on your notes, what do you think is happening in these 3 situations? Okay, so welcome back. I hope that you have actually jotted down some of your ideas or your thinking processes. So let's move on. So in chapter 6, we learned that all basic building blocks of a living thing is a cell. Okay, and plant and animals, you and I, we are examples of multicellular organisms. Now, Sec 1, similarly to the rest of the other lectures, if you need time to fill in the blanks, please make sure you pause the video. Okay, and you can pause because this is a recorded lecture. So you also learn in chapter 7 during your FHBL that matter is made up of 3 states: solid, liquid, and gas. And solid, liquid, and gas are actually made up of small, discrete particles. Now, the interesting thing about small, discrete particles is that these particles have the ability to move. Solid particles, although they are held together by strong attractive forces, they can still move. The only difference is that they do not move freely, but they vibrate about their fixed positions. Whereas if you notice, your liquid particles are able to slide past one another, and your gas particles are basically moving in all directions randomly at high speed. Okay, now since you and I are multicellular organisms, we are made up of more than one cell. We are made of millions of cells. How do our cells communicate with each other? How do our cells allow nutrients like sugars and fats and protein molecules to enter our cells to be stored in our vacuole? There must be a process by which these particles are able to enter our cells. And the third thing that you need to know is that our cells are held together by a partially permeable membrane. They are surrounded by a membrane. Now, in your previous chapter, you know that a partially permeable membrane is a membrane that prevents certain substances from entering, and it also prevents certain substances from exiting. Now, why is that so? Okay, so you may want to highlight this. So every time you see this blue box appearing, okay, it means you must take out your highlighter or you take out a different colored pen and put a star here to take note. Okay, now most of you would have been learning FCE for the first term. You would have used a sieve. Now a sieve is a utensil that you can find inside the kitchen that allows you to drain water from something, right? And why it's able to allow water to flow through? Because it's got small little holes. So similarly to your cell membrane basically has got tiny pores. Okay, so it has got tiny pores, and these tiny pores basically allow you, okay, to allow certain substances to pass through, but preventing substances that are too big to pass through the holes to actually exit the cell. So a partially permeable membrane, why is it partially? It's because it's got tiny holes that will prevent the entry or the exit of large substances or large particles. All right? Okay, so why is the movement of particles, all right, into cells so very important? Now, the movement of particles into cells are important because number one, cells require nutrients for growth, reproduction, and repair, okay, of damaged cells or damaged tissues. Next thing, just like your plant cells and your animal cells, why must they have vacuoles? Vacuoles basically allow them to store water. Now, water is very important. Water is important for hydration, for chemical reaction, and water is also very important because it's part of the process, it's involved in the process of photosynthesis in plants. Now, your cells also require vitamins. Okay, your body requires vitamins because vitamins need to be present to keep your defense systems healthy and to maintain bodily functions. So therefore, these particles, nutrients, water, and vitamins, they need to be able to move into cells. Now, why must they move into cells? Because cells require these particles for growth, reproduction, maintaining bodily functions, keeping defense systems healthy. Now, they basically move through two main processes, and these two main processes are known as diffusion and osmosis. So today we are going to be focusing on diffusion, and the next lecture we will be introducing ourselves to osmosis. Okay, so why is diffusion and osmosis so important? Now, when you breathe in, you actually take in oxygen from the atmosphere, and when you breathe out, you're actually removing carbon dioxide, which is a toxic gas. So oxygen enters your nose, but it doesn't stay there. It has to go into your body cells, right? So how in the world did it come from your nose, from the atmosphere to your nose, into your lungs, and into your blood. So diffusion is a process that is very important for the movement or what we consider gas exchange between your blood and your body cells. Okay, so in this picture, you can see that your lungs actually help to take in oxygen, and oxygen will then enter your red blood cells. Okay, and carbon dioxide will then be removed from your blood to your lungs, and when you breathe out, carbon dioxide actually comes out. So transport processes like diffusion and osmosis is very important because useful and waste substances need to be transported between your cells in living things, in our body and even in plants. Oxygen enters the blood from the lungs. Carbon dioxide leaves the cells and goes into the lungs to be removed. All right, so this is a very, very important process which we know as gas exchange. Now, this gas exchange also takes place in plants. All right. All right, it also takes place in plants. And one thing that you would like to know, so when we talk about gas exchange, we're talking about oxygen in cells and blood, going into cells in blood, and we're talking about cells and blood, okay, removing CO2. All right, so oxygen goes in and CO2 goes out. Whereas in a plant, it would be the opposite. In a plant, CO2 goes in, but oxygen goes out. So gas exchange actually relies on one of these two processes, which we will figure out later, which is the process that will be involved in this gas exchange. All right, another thing, the second thing about why these two processes are important is, can you remember in chapter 4, when we learn about separation techniques, we actually learn the production of new water from sewage water, right? From water that is dirty and chemically untreated. So what happens is that in new water production, they actually use reverse osmosis. Now, reverse osmosis means that they will use the process of osmosis, but the opposite way, all right, to actually get clean water from dirty water. Okay, so we can see that these two processes are very important, not only in living things, okay, I put here LT, living things, but also in non-living systems such as industrial applications. So these two processes that we will learn now, and you will also learn more of it in biology, is actually very applicable to daily life. Right? So let us move on. Okay, so you might take a highlighter and take note of this very important process. All right, so now let's start on the first process that we're going to be talking about, known as diffusion. Now, I'm sure all of you would experience this every day when somebody is cooking in the kitchen at home and you are in your room. You will notice that you can smell whatever that is being cooking in what is being cooked in the kitchen. You can actually smell the vapor particles, the scent particles from your room after a while. Why is that so? Even when you walk down from your classroom, okay, so whether is it the second level or even the first level, if you walk towards the canteen from the art circle past the musical staircase outside the action theatre, even before you queue up for your favorite canteen food store, you can already smell the food, the scent of the food, right? So where did the food come from? From the canteen, but you were quite some distance away, right? So why is it the smell is able to reach your nose? Okay, let's have a look at this. So let's pretend that this is the kitchen. You can see the number of particles here is a lot, right? Versus here. So therefore, we say that the number of particles found in the kitchen is the most concentrated. Now, what does most concentrated actually mean? Most actually means that it has a lot. Okay, so you can tell that there is a lot of particles here versus your room or your class. So if I were to circle, just in this circle, I can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. There are 22 particles here versus only 2 particles. Let's say this is in your room, okay, or let's say this is your classroom and this is the canteen. So do you notice how the particles were moving? How come you can smell the food from your classroom, from the canteen? It seems to be moving from the canteen all the way to your classroom. This is the movement of the particles. So I think you can predict that the smell seems to be doing something. It seems to be spreading, right? So there seems to be a spreading of smell. And why do you think the smell can spread? Let's say the scent or the smell actually has got particles, right? So what does it tell you about the particles? Were the particles stationary? Were they stationary or were they moving? Good, some of you can guess correctly. They were actually moving, right? So the spreading of the smell is actually due to the movement of the particles. And you will notice that they were moving from the canteen or your kitchen, which is the most concentrated because that is the source, or that is where the smell was coming from. Okay, and they were moving to your nose, which is not where the smell is coming from, right? You were not cooking in your nose, right? So we will say that it's moving from the most concentrated to the least concentrated. Now the word concentrated just means how many particles. So it's moving from an area that has got a lot of particles to an area that doesn't have that many particles. And this process of spreading is known as diffusion. All right, so this is the first process that we're going to be talking about. Okay, so can you please take out a highlighter and highlight this second point here? This is actually very important, that in diffusion, the spreading of the particles is because the particles are actually moving from an area that has got a lot to an area that has got very, very little. All right? So now let's delve deep into what diffusion actually means. Diffusion is actually the net movement. All right, so movement, remember, because chapter 7, we know that solid, liquid, and gas particles can move. It's just the extent or how much do they move, all right? Or how well can they move. So diffusion is the net movement of particles, all right, from a region of, can you guess that word, starting with the letter H, a lot of particles? Good. From a region of higher concentration to a region of lower concentration. So I just want you to imagine when you climb up a slide in the playground, okay? So if you're at the top and this is the bottom, okay, you actually slide down, or when you are cycling up the hill, and then when you go down the hill, you notice that it's easier to cycle down the hill versus cycling up the hill, right? So particles or objects have a tendency to fall quicker, all right, when they are going from a top to a bottom, all right, versus you trying to roll something from the bottom to the top. So same thing, particles tend to move from an area that has got a high concentration to an area that has got a lower concentration. I would like you to have a look at this animation. So you can see that when I press your perfume, how come after a while you can smell the perfume? Look at the perfume particles, the scent particles. It seems to be spreading throughout, right? And it's spreading throughout and evenly. Okay, so we say that diffusion spreads the particles evenly throughout the space that it's available. So eventually, after a while, you realize that the scent particles are all around because the spread is very even. So just like this video over, just this picture over here, even though you may drop the dye at the top, and you saw in the previous video, right, it sort of like moved throughout the liquid. After a while, the entire beaker will be blue, because the dye particles will spread evenly throughout the entire space. And diffusion can only occur when there is higher and lower. That means there is a difference. So you may ask, Miss Lee, so that means if there is no higher or lower, both places A and B got the same concentration, can diffusion occur? The answer is no, because there is no difference in concentration. Okay, so let me give you an example of what I actually mean. For example, I give you two areas, area A and area B. Area A is, let's say, your hall, the school hall, and area B is the canteen. Let's say the canteen has got 5 scent smell particles. The hall has got 0. There will be diffusion, because this is an area of high versus area here low, because here 0, here got more. But let's say these two places now, okay, let's say the hall also has got 15, the canteen also has 15, there will not be any diffusion because both numbers are the same, so there is no difference in concentration, then diffusion will not occur. But will there still be movement? The answer is yes, because all particles are able to move. All right, so that is something that we need to take note of. Okay, so what do we say? We say that diffusion only occurs when there is a difference in concentration. So I want you to turn your attention to this picture over here at the bottom. You can see that concentration, you can imagine concentration like the height. So the higher, the taller something is, and the shorter something is, that means there is a greater difference. Okay, so over here you can see that point A is taller than point B. So there is a difference in height. And because there is a difference in height, we can see that there's a difference in concentration. Point A having more, that's why it's taller, versus point B having lesser, that's why it's shorter. So the molecules or the particles will actually move down this particular concentration. And this difference in concentration, in science, we call this a gradient. Okay, so difference in concentration is also known as a gradient. In maths, when you draw a gradient is a difference between 2 points. So if the 2 points are exactly the same, then we say there is no gradient. But if one point is here and one point is at the bottom, and it looks like there's a slope, right? Then there is a gradient. Okay, so we say there is a concentration gradient because gradient actually means difference. Okay? Okay, so let's move on to the next one. So please take out your highlighter and highlight. The first thing is that diffusion requires a difference in concentration. Okay, so now let's move on. I would like to show you this animation. Now, you can see over here, there are 20 green particles. On this side here, there is 0 particles. Now, what is in between here? We say that this in between is actually like a divider. Now, I'm going to remove the divider. How do you think the particles are going to move? Are they going to move from this region where there is 20 to 0? Or are they just going to stay stationary? According to diffusion, it should move from the left-hand side to the right-hand side. So let's have a look. So you can see that it actually moves evenly throughout, and you can see that it slowly becomes equal 10, 10, right? So then you notice that the particles will start to move again, and then after that it will become 10, 10 again. So it will move until the concentration is the same. Okay? Alright, so now let's turn to the bottom part of page 3. We must take note of 3 principles in diffusion. Now, what is the meaning of principles? It just means rules. Okay, so there are 3 rules that must occur in diffusion. The first rule is the greater the difference in concentration, that means the more the difference is, okay, we say that diffusion will occur faster. Rate just means speed. So if the difference is not so great, then diffusion speed will be slower. If the difference is very, very great, that means the gap is very, very big, then we say that diffusion will occur at a faster rate. I just want you to look at this example. Now let's imagine that you are at a playground and you're looking at 2 slides, slide A and slide B. I want you to answer this question. Which slide do you think will allow you to go down faster? Which one do you think you will roll down faster? Slide A or slide B? Good, some of you predicted slide A, right? Why is that so? You realize that slide A is higher above the ground. You can tell that slide A, if I were to use a triangle, you can see that slide A is a steeper triangle, a triangle that is very slanted, right? But if you look at slide B, you look at the slope of slide B, it's a very gentle slope. So imagine you're at the top here on a bicycle, and if you roll down slide B versus rolling down slide A, obviously you will be traveling at a faster speed in slide A versus slide B. So why did Miss Lee talk to you about slides? I just want you to imagine that the slide, the height of the slide or the angle of the slide, okay? I think you all have learned angles already, right? So if you were to look at the angles here, okay, Miss, look at this. If I were to draw this as an angle versus this as an angle, which angle is actually greater? A or B? A, right? So if the angle is greater here, that means the slope is actually steeper. So you travel down faster. So I want you to just imagine the analogy is the height of the slide is very similar to the concentration gradient. Now remember, gradient is just another word for difference. Now in the exam, you don't have to use the word gradient. You can just say difference, and that is also the same. Okay, so you can see that there are more yellow particles here versus green particles here. Okay, so if you were to measure, this one has got 3, and this one, let's say you do a rough estimation, let's say this has got 25. This one, let's count, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 1, 2, 3, 4, 5, 6. There are 6. What is the difference? The difference is only 7. So it is not so high. But look at the difference here. 25 minus 3. 22. So imagine there are only 7 steps here, but there are 22 steps. Which one seems to be taller? This particular slide, right? So therefore, we say that the concentration gradient here is very steep because the angle here is very steep. This angle seems to be lesser. Okay, so it's less steep. So we say that the first concentration here in A, the concentration is greater, and therefore diffusion will occur at a faster rate. So just remember, greater, faster, lesser, slower. All right? So we can confidently say that concentration gradient or difference, all right, is similar to the height of the slide. So the higher the slide, the greater the gradient, the faster the diffusion rate. Now, the second principle, which we will talk about, is related to chapter 7. I just want you to look at this animation over here. Now compare, the solid particles are moving, but look at the way they are moving. They are vibrating about their fixed position. Okay, whereas if you look at the liquid particles seem to be moving as a cluster, but they have more freedom. But look at gas. It seems to be moving randomly and very quickly. So therefore, if I were to ask you to compare, which particles do you think will diffusion occur the fastest in? Remember, diffusion is movement. Which of the particles here seem to be moving faster? And I believe your answer will be gas. So, diffusion, the second rule is that diffusion occurs very quickly in gases. It can also take place in liquids, but it will be the fastest in gases because gas particles move the fastest. And since gas particles move the fastest, we can say that diffusion is the fastest in gas. So please take note of this, that gas particles move the fastest. Okay? So this is your link to chapter 7. Now let's move on to the third and final principle or rule of diffusion, and that is diffusion does not require a membrane to take place. Does it mean that diffusion cannot occur if there's a membrane? No. Diffusion can still occur. Diffusion can occur in the presence of a membrane if the particles that are moving through the membrane, just like this sieve here, is small enough to pass through the holes. If the particles are small enough to pass through the holes, okay, just like my red ink particles here, if they are small enough to pass through all the holes here, then diffusion can occur in the presence of a membrane. But if the particles are very big, they cannot pass through the membrane, we will say that diffusion does not occur, even if there is a difference in concentration. Okay, so I think it would be easier when I go through this example with you. Okay, so let's have a look. So when the particles are small enough to pass through the holes of the partially permeable membrane, we say that diffusion can occur in the presence of a membrane. Does diffusion require a membrane to occur? No. Let me give you an example. When you spray deodorant or perfume on yourself, okay, you notice that you can actually smell it from outside your room, but there is no membrane between your room to your living hall. So how come diffusion can occur? If diffusion cannot occur without a membrane, okay, that means it must occur with a membrane, then you won't be able to smell anything outside your room. And when you are walking to the canteen from your classroom, there's no membrane in front of you, yet you can still smell because diffusion can occur. So let's look at this example over here. On the left-hand side, All right. On the left-hand side, you can see you can see 1, 2, 3, 4, 5. There are 5 starch molecules and 0 starch molecules. Okay, this is side A and side B. Based on diffusion, starch will move from A to B. Why? Because starch has got more on side A and side B, right? So starch has got a higher concentration in side A versus side B, which is lower. In fact, it has got no starch particles at all. But why is it starch is unable to pass through the membrane? Look at the size of the starch particles versus the size of the pores. The starch particles will get stuck, so they cannot pass through. So we say that starch molecules are too big to pass through, so they cannot diffuse, okay, to side A. However, now, if you look at the glucose molecules here, which are your smaller molecules, what do you notice about the size of the glucose molecules? Are they small enough to be able to pass through? The answer is yes. And since side A also has higher concentration of glucose, side B only has 1, 2, 3, 4, 5. So obviously, glucose will move from A to B. And why can glucose move? Because glucose is small enough to diffuse across. Right? So this actually explains to you that diffusion does not require a membrane, but when a membrane is present, then the particles must be small enough to diffuse. If not, no diffusion will occur. So we've seen the 3 rules on page 3. Now let us apply our new knowledge to 2 questions. All right? All right. So take out your highlighter, highlight this point. You also want to highlight that starch is too big, whereas glucose is small enough. That's why diffusion of glucose will occur, but diffusion of starch will not occur because starch is simply too large, too big, too huge. You'll get stuck. Okay, so let's look at this. We know what this is already. Let's say you put perfume, you go to Sephora, okay, or you go to Robinsons, or you go to a shopping center, Isetan, and when the person sprays the perfume, you can smell it, right? Even though you are at least 2 to 3 meters away, because there is diffusion of the vapors to other parts of the room. All right, and if you drop a red ink at the top of the cup, you will notice that the red ink will actually diffuse and spread evenly throughout the entire glass of water until the whole liquid becomes red. Okay, so this is what you will see. You will notice, then you will notice that the blue ink, in this case here blue, it spreads throughout the liquid. All right, now let's look at question 1 at the bottom of page 4. Now let's look at this experiment over here. It says that the diagram over here shows an experimental setup which is used to investigate diffusion. Now there are a few things that I would like you to take note. Whisking tubing is actually a partially permeable membrane. Please write that down. This is actually your whisking tubing. So how do you know it's a partially permeable membrane? You can see that there are tiny holes that are found on the surface of the membrane. Okay, so there are holes in the surface of the membrane. Now the starch solution is inside the whisking tubing. So this is your starch solution. Iodine is a reactant, it's a chemical reagent, and I'm going to use yellow color to represent. It is actually outside the whisking tubing. Now, the original color of iodine is actually yellow-brown. You will see a video later, okay? This is yellow-brown. Now, when iodine comes into contact with starch, that means if starch is present and iodine is present, iodine will actually turn blue-black. Okay? If iodine remains yellow-brown, that means starch didn't react with it. Okay? So, this is the test tube. That means iodine is actually outside in the test tube. Alright? Then, the starch is actually inside the whisking tubing. So, this is starch. And this is iodine. So I want you to compare, the first question I want you to compare is which molecule do you think can pass through the membrane? Look at the size of the hole look at the size of the hole, and look at the size of the molecule. Which molecule is small enough to pass through? I think it's quite obvious. Okay? The second question I want to ask you is this. If this molecule can pass through, which, where do you think will turn blue-black? The inside of the Visking tubing, or you think the outside will turn blue-black? Okay, so before you answer my questions, let's have a look at a video, a simple video that demonstrates this experiment, all right? apparatus is turning blue-black. You notice that it's only the inside of the Visking tubing that is turning blue-black, whereas the outside is actually still remaining yellow-brown. So that means starch didn't come out of the Visking tubing, because if starch did, then you should be able to see the outside turning blue-black. And you look, after a few hours, the iodine solution outside became lighter, right? That means iodine actually moved in. Alright, so I ask you, what type of membrane will pass through the partially permeable membrane? Will it be molecule A or molecule C? Yeah, molecule A, which is your iodine molecules, they are small enough to pass through. And because they are small enough to pass through, only your iodine will enter. And when your iodine enters, inside here, which has starch, will actually turn blue-black, because iodine is now reacting with starch. But did your starch molecule actually move out? It didn't move out, right? And if it didn't move out, outside has got no starch. And if there is no starch, iodine will remain yellow-brown. So what is the identity of molecule C? Okay, molecule C is your starch molecules. And that is why we say that the outside didn't become blue-black. You saw, right, in the video, it was only the inside of the tubing that became blue-black. Okay? Alright, so let's move on to the next question. Which of the following descriptions about diffusion is correct? Alright, so let's have a look. Alright, so which of the following of descriptions of diffusion is correct? Does diffusion require a partially permeable membrane? The answer is no, it does not require a partially permeable membrane. Obviously, your answer C and D will be out. So how does diffusion move? So diffusion moves from, remember your slide, How does diffusion move? It doesn't go from, you don't go up a slide, right? You actually have a tendency to slide down, right? So we say that it moves from high to low. So your answer for this question will be A. So please take down your answer. And we will end this segment by looking at a video, okay, by looking at animation that basically summarizes what diffusion actually is. So you can see that diffusion is the spreading of particles from an area that has got very high concentration to the area that has got lesser concentration. B, we also, right. So this brings us to the end of our lecture. In the next lecture, we will actually be talking about the examples of diffusions in plants and animals. And then we will then talk about the second transport process, which is known as osmosis. Okay, so till then, thank you very much and have a fruitful HBL day.